All right, today we're diving deep inside our very own cells to explore some absolutely incredible microscopic machinery, the master untanglers that keep our genetic code from tying itself into an impossible knot.
So let's kick things off with a mind-bending puzzle that's happening inside every single one of your cells right now. Get this, if you took the DNA from just one of those cells and stretched it all the way out, it would be about 6 feet long. Yeah, 6 feet. So how in the world does all of that get packed into a nucleus that is thousands of times smaller than the head of a pin?
And here's the kicker. It's not just about storage. It's not like you can just cram it all in there like you're stuffing a sleeping bag away for the winter. No way. The cell has to be able to read this incredibly dense library of information all the time. For things like replication and transcription to happen, the machinery has to get in there, unwind the DNA, and wind it back up, all without creating a catastrophic tangled mess.
So how does the cell pull off the seemingly impossible challenge? Well, it calls in the experts. The answer lies with a set of truly elegant molecular machines.
It uses this family of enzymes called DNA Topoisomerases. You can think of them as tiny molecular surgeons, or maybe even master locksmiths for our genetic code. Their entire job is to manage the shape and structure, the whole topology of DNA by cleverly cutting, rearranging, and then perfectly sealing the DNA strands back up again.
These amazing enzymes, first shown to exist by the biologist James Wang, are totally fundamental to life as we know it. They are literally everywhere, from bacteria all the way up to humans, and they work tirelessly to relieve all that twisting stress, it's called supercoiling, that builds up in our DNA whenever it's being read or copied.
Okay, so let's dive into how they actually work. Topoisomerases have two main strategies for managing DNA. And you know what? It all comes down to a really simple difference: the number of cuts they make.
So you've got your type 1 enzymes and your type 2. Type 1 makes just a single cut in one strand to relieve tension. And what's really cool is it cleverly uses the energy that's already stored in the twisted DNA. But then you have type 2, which performs a much more dramatic move, a double strand cut. And that's such a complex operation that it actually requires a dedicated energy source, which is ATP.
And even within that type I family, you've got different styles. Think of type IA as doing a little magician's trick. It cuts one strand and passes the other one straight through the break before sealing it up. Then there's type IB, which is more like a controlled release valve. It cuts one strand and lets the entire DNA molecule spin around the other strand to unwind before it reconnects it.
Now for the really intricate stuff. The type 2 enzymes perform a kind of molecular surgery that is just astonishing. They solve the problem of two tangled sections of DNA by creating what's called a DNA gate, which allows one part to pass directly through another. So let's break down how they pull this off.
It's this brilliant 6-step process. First, the enzyme grabs one DNA duplex, that's the gate segment. It then makes a clean cut right through both strands. After that, it captures a second, transport, segment, and passes it right through that gate. It uses ATP energy to close the gate behind it, and finally, it perfectly reseals the original break. It's just an incredible feat of molecular engineering.
So with these mechanisms, topoisomerases have this whole toolkit of topological tricks. They can link and unlink separate DNA circles, a process we call catenation and decatenation. They can literally tie and untie knots in the DNA. And all these maneuvers are absolutely essential for keeping our genetic code organized and accessible.
Now, this is all fascinating biology, but here's where it gets really interesting for us today. This fundamental cellular process has become a critical target in modern medicine.
Think about it. If a cell can't manage its DNA, it can't divide. It's as simple as that. So if you can jam this essential untangling machine, you can stop things like a bacterial infection or the runaway growth of cancer cells right in their tracks.
And this is exactly how a lot of our modern drugs work. Common antibiotics like ciprofloxacin, they specifically target the type 2 enzymes in bacteria, which kills them without harming our own cells. In the same way, powerful chemotherapy drugs work by targeting the topoisomerases in our own rapidly dividing cancer cells, stopping them cold.
The way these drugs work is really clever. They don't just block the enzyme, they actually act as poisons. They let the topoisomerase make its cut in the DNA, but then they lock it in that state, preventing it from resealing the break. This creates lethal breaks in the DNA, which triggers a cell to stop its cycle and ultimately self-destruct.
But this story is far from over. This is a field where some of the biggest questions are still just waiting to be answered.
So, let's just recap the key takeaways. We've seen how topoisomerases solve DNA's massive packaging problem.
We know they're classified into type I and type 2, based on making single or double cuts. They perform all these critical reactions like unknotting our genetic code. And because they're so essential for life, they've become this huge target for medicines like antibiotics and cancer drugs.
And that leaves us with a really fascinating final thought. Despite everything we know about these incredible molecular machines, we're still just beginning to understand their full complexity and elegance. How do they know exactly where and when to cut? How do they achieve such precision without making catastrophic errors? These are the questions that are going to drive the next wave of discovery.